Hello and welcome back. We are still in Chapter 1 Biodiversity, but in this video, we're going to enter into the final subtopic. So if you'd please, join me as we enter into 1.7 Domain Eukarya, Kingdom Animalia, which is what we're most familiar with because these are our animals. Now, before we go into the lesson, as per usual, let's have a look at the learning outcomes for this subtopic. I hope that by the end of this lesson, you should be able to describe the unique characteristics of organisms in Kingdom Animalia and also state the classification of Animalia into 9 phyla and the 9 phyla as stated here. And also, I hope that you're able to discover the unique characteristics of the 9 phyla. Now, I'm going to separate this very long subtopic into several videos. In this video, we're only going to look at Learning Outcome 1.7a. So we're only going to be looking at the unique characteristics and have an overview of the 9 phyla. After you're done with this video, you're going to watch each of the 9 videos that I have for each of the 9 phyla. So if you're ready, let's continue. So let's talk about the unique characteristics of this most familiar kingdom of organisms. And you can look at yourself for each of these and see if they apply to you. First things first, Animalia are heterotrophs. They are multicellular eukaryotes. They lack cell walls and they have a period of embryonic development. So far, this is true for all of us. Let's have a look at this thing about embryonic development. What does it mean by period of embryonic development? This means that when the sperm and egg fuse and form a zygote, it will undergo a stage as an embryo before it is born or hatched and continue with the rest of its life. So imagine when you were being fertilized and you became a zygote, you had to go through 9 to 10 months of gestation in your mother's womb as an embryo, as a fetus, before you were born, and then you can start your own life as an independent individual. So other animals will have this period of embryonic development as well, either in the womb, if it's a mammal, or in an egg, etc., etc. These Animalia are usually motile, and most of them, if not all of them, are motile, and they have nervous and muscle tissue. So kita ada cell-cell saraf, okay? Dan kita ada cell-cell otot. They are diploid, which means they live a diplontic life cycle. You and I, we have 2 end in most, if not all of our cells. And I hope that as we go through this list, you notice that all of this has applied to you. All right, now we're going to go into this next part where we talk about the evolution of animals. So as I have shown you in the history of the world, I guess, video, life started as microscopic specks, and over millions and millions of years, it has built from unicellular, eventually becomes multicellular, and when it's multicellular, it takes on many shapes and forms. Jadi kalau kita imbas balik evolusi kita dari mulanya kehidupan di bumi sampailah sekarang, kita boleh nampak ada trend, okay, perkembangan evolusi haiwan-haiwan, tumbuhan dan sebagainya. So bila kita tengok evolusi haiwan, kita tengok beberapa criteria. When we're looking at the evolution of animals, we look at 4 main criteria. The first of these criteria that we will look at is whether or not the organism has body symmetry. Is it an asymmetrical body plan, a radially symmetrical body plan, or a bilaterally symmetrical body plan? Okay, so when we look at that, we can see, okay, how far evolved is it from the very first animal? Another criteria that we could look at is the body plan of the organism. So does the organism have a diploblastic body plan or a triploblastic body plan? A diploblastic means it has 2 germ layers when it is forming the blastula stage, and the 2 layers are endoderm and ectoderm. We'll look at this more later. If it's triploblastic, means it has 3 germ layers, which is the endoderm, mesoderm, and the ectoderm. The third criteria that we will look at when we study the evolution of animals is its body cavity. So we look at whether it is acoelomate, pseudocoelomate, or coelomate. If it is coelomate, it can be divided into protostomes or deuterostomes. Protostomes are organisms where the blastopore, the first hole that forms when it is in the embryonic development, when the first hole becomes the mouth of the organism, that organism is called a protostome. If the first hole of that embryo becomes the anus of the organism, then that organism is classified as a deuterostomes. The blastopore is the first opening of the gastrula cavity and that will later on develop into the gut of the organism. Kalau ikut syllabus kamu yang terkini, dia lebih tekankan kepada body cavity yang acoelomate, pseudocoelomate dengan coelomate sahaja. Syllabus lama dia lebih tekankan kepada protostomes dan deuterostomes. Jadi bagi yang XPDT kamu ada belajarlah pasal ni. Tapi mungkin ada buku ataupun nota yang sekarang dia tidak berapa tekankan protostome dengan deuterostome. But actually this is quite an important thing if you want to go and learn more about animal diversity or animal evolution in the future. You should know about protostomes and deuterostomes. Okay, now we move on to the fourth criteria that we look at when we study evolution of animals, which is segmentation. Is the body segmented or unsegmented? Now let's have a look at each of these criteria in more detail. First, we'll have a look at body symmetry. When we talk about body symmetry, is it asymmetrical, which is have no pattern of symmetry, or is it radially symmetrical? So if an organism is radially symmetrical, cutting through the center of the structure in any direction will produce two halves that are mirror image to each other. Contohnya, kalau kita potong ini sea anemone, okay, kita potong dari mana-mana arah pun dia akan bentuk mirror image. So, kiri dengan kanan, atas dengan bawah adalah sama. Whereas if it's bilaterally symmetrical, the body can be divided into two halves that are mirror image to each other if cut along one plane only. So, ini kalau kita belah separuh sahaja, satu arah sahaja, baru kita boleh dapat symmetry. Okay, so that is it for body symmetry. Now we shall go on to the next criteria, which is body plan. So when we talk about body plan, we are going to cut the organism in half and then have a look at its tissue. Okay, kita akan potong organism tu, kita tengok berapa layer germ cell yang ada pada organism ni. So kita biasanya tengok dari segi dia punya adult stage dan boleh juga kita tengok dari segi dia punya gastrula stage. But mostly it is from the adult stage. So if the organism is diploblastic, it's only going to have two layers of germ cell. That means in the embryo, it forms ectoderm and endoderm. Lepas tu kawasan yang di tengah-tengah itu akan jadi dia punya gut ataupun dia punya saluran pemakanan daripada mulut sehingga ke lubang dubur. Okay, so as it develops into adult, it becomes ectoderm, a mesoglia, and endoderm. Mesoglia ini macam satu lapisan tisu, tapi dia bukan sel yang hidup lah. It's a non-living layer, and it's going to separate ecto and endo, but it doesn't count as its own tissue layer. For triploblastic organisms, you can divide it into three more body plans, which is the acoelomate, no coelom. Coelom adalah macam ruangan kosong di antara germ layer. Okay, so this one, it has ectoderm, mesoderm, endoderm. Cuma, macam kamu nampak di sini, dia punya ectoderm, mesoderm, endoderm semua melekat sama-sama. Tiada ruangan kosong di antara germ layer dan ini dikira sebagai acoelomate, tiada coelom. The second body plan under triploblastic germ layers is pseudocoelomate. The coelom is not completely lined by the mesoderm. So here we have the blue one, which is ectoderm, mesoderm, endoderm macam terapung di dalam ruang kosong itu. So ini dikira sebagai pseudocoelom. The third one is coelomate. So this one, there is ruang kosong, but the ruang kosong is surrounded or lined by the mesoderm. So the blue one is ectoderm. The red pink one is the mesoderm. and the yellow one is the endoderm. As you can see, the coelom is here, surrounded by the mesoderm. Okay, so that is how you remember between these three triploblastic patterns. Now we're going to continue with body cavity. When we're talking about body cavity, it is still going back to that coelom stuff. When we talk about it, it is talking about a fluid-filled cavity that forms within the mesoderm. The function of the coelom or body cavity is to cushion the internal organs and to prevent internal injury. So mengelakkan kerosakan ataupun melindungi kita punya organ-organ dalaman. It also can act as a hydrostatic skeleton for organisms that do not have heart, bones, or exoskeleton like us. So macam cacing-cacing, dia akan gunakan dia punya coelom sebagai dia punya hydrostatic skeleton. And it also enables the internal organs to grow and move independently of the outer body wall. So organ-organ di dalam dia boleh bebas untuk membesar, develop tanpa dikongkong oleh lapisan tisu luar. Okay, so if it's acoelomate, it's like this. Pseudocoelomate is like this. Coelomate, it's like this. Acoelomate organisms have a solid body and lack a body cavity. For pseudocoeloms, they have a body cavity that is not completely lined by the mesoderm. A coelomate is an organism with a fluid-filled body cavity completely lined by the mesoderm. Okay, and then we go on into the fourth criteria, which is segmentation. Okay, fourth criteria, segmentation. So we look at whether or not the organism is separated into segments. Okay, kalau dia segmented means dia ada dibagi kepada beberapa segmen. Or if it's unsegmented, like this nematode, okay, nampak cacing ni, dia licin saja dari mula kepada akhir. Cacing ni pun licin saja, mula kepada akhir. So, ini tiada segment. Tapi kalau kita tengok cacing tanah, we look at this earthworm, it has segmentation. Setiap bahagian dia ada nampak dia punya garis-garis. Flies and most insects also have a very obvious segmentation on their body. Sometimes the segment is big, like the whole head or the whole thorax, and sometimes it is separated into abdomen. So, tidak semestinya satu badan tu ada garisan baru kita kira segmented. Kadang-kadang kalau kita bagi kepada kepala, thorax, dan abdomen pun sudah dikira sebagai segmentation. Right, and then as you may already be aware, we can classify organisms in kingdom Animalia into 9 phyla, which is phylum Porifera, phylum Cnidaria, phylum Platyhelminthes, phylum Nematoda, phylum Annelida, phylum Arthropoda, phylum Mollusca, phylum Echinodermata, and phylum Chordata. So each of these phyla will have its own short video, and I encourage you to have a look at them. And in each of these videos, I will also link to National Geographic or other documentary style videos, which have much nicer pictures with better video quality. So that's it for this segment. But before I can tell you to watch the other videos, I would first like to clarify how we can classify organisms in kingdom Animalia into these 9 phylum. So we start with the ancestral protista. So the very first animal was a protist that eventually became multicellular, and over millions of years of evolution, it's going to divide into 2 types of animals, which is animals with no true tissue and animals with true tissue. Animals with no true tissue are classified under Parazoa. With true tissues, we classify it under Eumetazoa. Parazoa means dia macam haiwan tapi macam tidak kira haiwan juga, tapi macam haiwan juga. So kita classify dia sajalah under kingdom Animalia tapi kategori asing. Eumetazoa, Eu means true, metazoa, true multicellular animals. So Parazoa will go into phylum Porifera. Kalau Eumetazoa, we look at other criteria. Under Eumetazoa, we look at whether it has a radial symmetry and diploblastic, or bilateral symmetry and triploblastic. If it's radial symmetry and diploblastic, it is we label it as radiata. If it's bilateral symmetry, triploblastic, we label it as bilateria. So radiata would bring us to phylum Cnidaria. When we look at bilateria, we look at another criteria, which is the body cavity. If it does not have body cavity, it is acoelomate. If it does have body cavity, it will go into the other criteria. Acoelomates are classified into phylum Platyhelminthes. If it has body cavities, we look at whether or not the body cavity or the coelom is lined by a mesoderm or completely lined by a mesoderm. If it's not completely lined by a mesoderm, it is under pseudocoelomates. If it does get lined by a mesoderm, it is under coelomates. So pseudocoelomates bring us to phylum Nematoda. Coelomates, we divide it into protostomes and deuterostomes. If it is protostomes, it can go into either of these three phylum, which is Annelida, Arthropoda, Mollusca. If it is deuterostome, then it goes into either phylum Echinodermata or phylum Chordata. Macam mana kita tahu mana daripada tiga ini, kita tengok lagi sama ada dia lebih mirip organism dalam phylum ini ataupun phylum itu. So we will have to study the unique characteristics of each phylum. Then we can take that organism and really classify it into the correct slot. So that's it for this segment of the lesson. Thank you very much for your time and attention, and I hope you go and watch the other videos for each of the 9 phyla. And I'll see you there. Thank you, and bye!